Don't forget to subscribe and hit the notification bell. What is microbiology? It is a study of bacteria, fungi, viruses, protozoa, and algae. It is a study of microorganisms that can't be seen by the naked eye. It is also called microbes or bacteria. Microbes are everywhere. They are forever underfoot, crawling all over places, and sometimes it's on the air we breathe. By using microscope, we are able to see the tiny organism. Compound microscope, also known as high power or biological microscope, they are used to view specimens not visible to the naked eye such as blood cells and microorganisms. Compound microscope typically includes 3 to 5 objective lenses that range from 4 times to 100 times, combined with 10 times eyepieces. Total magnification ranges from 40 times to 1,000 times. Stains are viewed after they are being mounted on the glass slide. When using 100 times objective lenses, immersion oil is used to concentrate light and increase resolution of the specimen image. Stains are useful for enhancing contrast in the specimen and therefore in improving in viewing experience. A mechanical stage is useful at higher magnifications. It enables you to move a slide easily back and forth as well as side to side. Because bacteria are colorless and usually invisible to light microscopy, colorful stains have been developed to visualize them. The most useful is gram stain, which separates organism into 2 groups: gram-positive organism, which stains purple, and gram-negative organism, which stains red or pink. This stains also allows the doctor to determine whether the organism is round or rod shape. Gram staining or Gram's method is an empirical method of differentiating bacteria species into 2 large groups based on the chemical and physical properties of their cell walls. The method is named after the inventor, the Danish scientist Hans Christian Gram, who developed the technique in 1884 to discriminate between pneumococci and Klebsiella pneumoniae bacteria. Gram stain are performed on body fluids or biopsy when infection is suspected. For any stain, you must first smear the sample to be stained onto the slide. After making the smear, then heat fix the bacteria on the slide using the incinerator. Incinerator is an infrared heat sterilizer. It is safe and simple to operate without fire and ideal for use in the field of microbiology. There are 4 steps on gram staining. First, pour on crystal violet and wait for 1 minute. A basic dye, crystal violet, is used to stain the slide. This dye is taken up by both gram-positive and gram-negative bacteria. The slide should look purple to the unaided eye, and if examined microscopically at this point, both gram-positive and gram-negative bacteria are purple. Second, wash gently with tap water for a maximum of 5 seconds and flood with iodine solution. Let it sit for 1 minute. This acts as a mordant and fixes the dye. Wash off again with water. Add the colorizer, either alcohol or acetone, several times to the slide, allowing to run off until the color turns clear. This washes away all the unbound basic dye, usually crystal violet, and leaves gram-positive organisms stained purple and gram-negative unstained. Finally, it's the 4th step. Counterstain with saponin, wait for 1 minute, and rinse with tap water. This stain is taken up by both gram-positive and gram-negative organism, but does not alter the color of gram-positive organism much, as they are already purple. It does, however, make the gram-negative organism pinkish red. Black dry or air dry the slide and examine under microscope. When the slide is studied microscopically, cells that absorbs crystal violet and hold on it will appear purple. These are called gram-positive organisms. However, if the crystal violet is washed off by the decolorizer, these cells will absorb the safranin and appears red. These are called gram-negative organisms. Organisms that cannot reliably be differentiated by this staining technique are said to be gram-variable. If you look at this slide, the organism is half pink and half purple. This is what you call gram-variable bacilli.